A nitrogenous base is simply a molecule that contains nitrogen and has the chemical properties of a base. Let's break it down a bit to understand what the definition really means. Nitrogenous bases fall into the class of chemical compounds known as organic compounds, or those which contain carbon. What makes a nitrogenous base is that it not only contains carbon, but it also contains the element nitrogen. Elemental nitrogen is normally found as a gas in the Earth's atmosphere. It is generally odorless, tasteless, and colorless, and reacts very easily with the other elements. This reactivity makes it an important part of the compounds necessary for life. Nitrogenous bases are also, as the name implies, bases. A base is a substance that can donate pairs of electrons to other elements or molecules and form a new molecule in the process. It is the opposite of an acid, which is a substance that can accept pairs of electrons from elements or molecules. In the case of nitrogenous bases, nitrogen bonds with carbon, hydrogen, and oxygen atoms. These different elements can arrange themselves in rings, and we name the compound based on its ring formation. Bases that form single rings, like this molecule, are called pyrimidenes. Bases that form double rings, like this molecule, are called purines. Purines are much larger than pyrimidenes because of this double ring. Purines and pyrimidenes serve as a basis for the building blocks of DNA and RNA. DNA, or deoxyribonucleic acid, is the molecule that encodes the instructions for life. RNA, or ribonucleic acid, is the molecule that is responsible for the coding, decoding, regulation, and expression of genes. RNA is single-stranded, whereas DNA is double-stranded. These instructions are encoded in the order of nitrogenous bases all along the DNA and RNA molecules. These bases are formed starting with either the single-ring pyrimidine or the double-ring purine. Then some extra nitrogen, hydrogen, or oxygen molecules are added on to the basic ring to make the nitrogenous bases: adenine, guanine, cytosine, thymine, DNA only, or uracil, RNA only. When bound to the phosphate backbone of DNA and RNA, the nitrogenous bases are called nucleotides. Cytosine and thymine are both pyrimidine bases. Cytosine starts out as the single-ring pyrimidine, and an extra oxygen, nitrogen, and 3 hydrogen atoms are attached to the ring. Cytosine is very unstable and can change form if left on its own for too long. Thymine also starts out as the single-ring pyrimidine, but has 2 extra oxygen atoms as well as 2 hydrogen atoms and a methyl group attached to the main ring. Uracil is also a pyrimidine base, but it is only found in RNA, not DNA. It is very similar to thymine except it does not have the methyl group, so it acts differently from thymine. Adenine and guanine are both purine bases. Adenine starts out as the double-ring purine, and an extra nitrogen and 2 hydrogen atoms are attached. Guanine also starts out as the double-ring purine, but has an extra nitrogen, 3 hydrogens, and an oxygen attached to the rings. In DNA and RNA, these nucleotides pair up. Pyrimidine bases with their single rings always pair with the double-ringed purine bases. In DNA, the pyrimidine cytosine always pairs with the purine guanine, while the pyrimidine thymine always pairs with the purine adenine. RNA does not contain thymine, though, so in RNA, adenine pairs with uracil. The nucleotides pair up to form DNA and RNA, and the order in which they pair up determines what an organism looks like and how it acts. DNA, or deoxyribonucleic acid, and RNA, ribonucleic acid, are the building blocks of life that either contain genetic material, DNA, or code, decode, regulate, or express genes, RNA. These building blocks are composed of a phosphate backbone and nucleotides, which are nitrogenous bases attached to the phosphate backbone. A nitrogenous base is simply a molecule that contains nitrogen and has the chemical properties of a base. There are 2 kinds of nitrogenous bases present in DNA and RNA: pyrimidine bases and purine bases. The pyrimidine bases are cytosine, thymine, in DNA only, and uracil, in RNA only. The purine bases are adenine and guanine. Adenine always pairs with thymine, or uracil, and cytosine always pairs with guanine.